Welcome to Agam's lecture series, a lecture series designed to bring learning to the comfort of your home. I remain your lecturer, Agama you Agam, and today we are still treating the subject General Biology 1, coded GNS 132 230, and the topic is genetics. We have already discussed part of this genetics in our lecture 8, where we discussed mitosis. Today we are going to continue that discussion with what? Meiosis and the rest of other aspects of genetics. Now, meiosis is a type of cell division that produces what? Gametes. It occurs in two successive divisions. Therefore, the prophase is marked prophase 1, Metaphase 1, Anaphase 1, and Telophase 1. The second set of the divisions is Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. Let us now look at the diagram of meiosis so that we know the details of what happens. But let me also say here that the details of what happens resemble the same thing that happens during mitosis. Now look at the diagram of meiosis. Prophase 1. During prophase 1, just like we said, the chromosomes will thicken, the chromosomes will become visible, and the chromosomes will pay up. During metaphase 1, the paired chromosomes will line up in the equatorial region, like we said before. During anaphase 1, the chromosomes have each pair of them, which is now called chromatids, and join at the center, will move to the opposite ends of the cell, which we call the opposite poles. During the telophase 1, two nuclei are formed, and it's like two daughter cells have been formed. These two daughter cells will rest briefly in a type of rest called interkinesis. And this phase 1 will now enter into this phase 2, which is called the second meiotic division. Note that the first one can also be called the first meiotic division or the first meiotic phase. Now we are now looking to the second meiotic phase, which will now start with prophase 2. Just like we said, chromosomes also will thicken, shorten, and become more visible. Entering metaphase 2, the chromosome will move to the equatorial region at the center of the cell and align themselves. During anaphase 2, the chromatids will now separate and move to opposite ends of the cell. Now, it's two daughter cells that started this. Now, during telophase 2, the two daughter cells, one will divide into two, and another one will divide into two, resulting into 4 gametes. Remember that the other one, mitosis, just produced 2 daughter cells, but this one is producing 4 gametes, and these 4 gametes will now take part in sexual reproduction. Remember that the gamete is haploid, while a normal cell is what? Diploid. Now, having said that, let us look at the significance of meiosis. Now, they produce haploid gametes, and I told you they produce 4 haploid gametes that take part in sexual reproduction. Meiosis gives rise to genotypic and phenotypic variation. It gives rise to what? Variation. If you can't even remember genotypic, so it gives rise to what? Variation. And also, this variation is the basis for natural selection in organisms. Now, we are also going to look at the differences between mitosis and meiosis. Now, mitosis, we said that this one occurs during growth and development, while this one occurs during the formation of what? Gametes. We said that in this one, 2 daughter cells are produced, but in this one, 4 gametes are what? Produced. We said in mitosis, the division is what? Equational. They produce equal daughter cells, but this one is what? Reductional. They reduce the diploid to what? Haploid. Now, during mitosis, there is no crossing over, that's exchange of genetic material. But in meiosis, there is exchange of genetic material, and that's what makes an individual that will result to resemble the father and the mother at the same time, because during the division there's crossing over of genetic material between the chromosomes. Now, in mitosis, it doesn't lead to what? Variation, but in meiosis, it leads to what? Variation. Now, we are now going to enter into genetics proper. We have now laid the foundation for the course of genetics. genetic terms. Hybrid, simply the offsprings of two different parents, two dissimilar parents. That's a hybrid. Now, allelomorph, a pair of genes that stays on the same locus on a chromosome and they control a pair of contrasting character. If you say that the character is contrasting, let's take a character example. Somebody's height. You can either be tall or short. So it's contrasting. Take on skin color. You can either be dark or fair. Take your nose shape. You can either be pointed or flat. Take your your eyes. You can either be brown eye color or blue eye color. So all characters has two versions of it, and that's what we make what contrasting what characters. You can call it a pair of alleles, or alleles, allelomorph, or a pair of what alleles. Then gene is the unit of what inheritance. We have already told you that this gene is made of contain chemical ingredient DNA, deoxyribonucleic acid, which is the chief executive of the cell. Even when there are dispute in the father of a child, you go for DNA test because all the information about the child is located in the what DNA. Then, if a pair of chromosomes are identical, like A, capital A and A, you say it is what homozygous. Homo means what the same. If the pair of chromosomes are not identical, including they are different, small capital A and small a, you say they are what heterozygous. Hetero means what different. Homo means what the same. Now, filial generation, the offsprings of a father and a mother, their offspring is what we call filial generation. First offspring, first filial generation. Second generation offspring, second filial generation. Third generation offspring, third filial generation. In that order, you continue going. Then you say dominant character or dominant gene. A gene, take for that, somebody is fair in color because the gene for fair color is dominant. If the gene for fair color is not, the person will not be what fair. Therefore, a dominant gene or a character is a gene or a character whose effect is shown, whose effect is what expressed, whose effect is what manifested. Recessive gene or character, a gene or character whose effect is not shown, whose effect is unexpressed, whose effect is what covered, whose effect is what masked, and is masked because there is a dominant gene. The hybridization, forming fusion of mixing of genetic material from parents, opposite what parents, like a father and a mother will produce a child. That act of fusion of their two genes is called what hybridization. And offspring of a hybridization is what a hybrid. Now, you now talk of phenotype. If you are light skinned, or you are tall, or you are short, or you have brown eyes, you have these brown eyes because the gene for those brown eyes are expressed. So phenotype is the expressed qualities by which an organism is what recognized. In a class, I can ask you, show me the tallest person in the class. You are seeing the tallest person because it's expressed in him. Then genotype, the total number of genes that possessed by an organism is what genotype. Those ones that you can see physically and those ones you cannot see physically. Those that are expressed and those that are not what expressed. The totality of all genes make up the what genotype. Mendelianism and Mendelian laws. Gregor Mendel developed the science of genetics in 1856, and after a series of experiments, he came up with Mendelian laws of genetics. We have two laws: law of segregation of genes and law of independent assortment of genes. The law of segregation of genes states that genes are responsible for the development of an individual, and that these genes occur in pairs, and these genes are transmitted from generation to generation without altering their form. That's why somebody can resemble the mother and later resemble the great grandparents, because these genes don't change their form as they move from generation to generation. Then you look at the second law, law of independent assortment of what genes. These genes separate independently and is transmitted independent of other what genes. Take for instance, the gene for your skin color, the gene for your height, the gene for your nose shape. The gene for your nose shape does not wait for the gene for the skin color to before expressing itself. Each one expresses itself independent of the other one, and that's why you can have qualities that make you ugly, qualities that make you beautiful at the same time, because one is not saying waiting for the other. Each expresses itself independently. Now, we're now going to look at Mendelian inheritance. We are going to demonstrate by using a father and a mother, but one is tall, one is short. Just very briefly, as we have a father here, I have a mother here, and they are hybridized. This is hybridization we are now doing, fusion of gamete from the two parents. This one is tall, and this one is short. You know, we are representing dominant one with capital letter and recessive one with small letter. So these are parent generation. For these two to reproduce, they will undergo what? Meiosis. Once they set on meiosis to produce gametes, so this one is a gamete, and this one is a gamete. This one is a gamete, and this one is a gamete. Now these have become our parent what? Gametes. If parents and gametes are produced, the next thing is for them to undergo what? Fertilization to produce young ones. This one will join with this. This one will join with this. This one will join with this, and this one will join with this. The result will now be capital T small t, capital T and small t, capital T and small t, and capital T and small t. This becomes the first generation offsprings. And you can see that they are all capital T and small t. So when you look at this, they all of them will appear what? Tall. Why are they appearing tall? Because tall is dominant to what? Short. So looking at them, they will all appear. But if you don't carry these offsprings of this first generation, they now become their own parents to produce grandchildren. You cannot see. Just watch for what are they. This one becomes a gamete here. 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 And this gamete will now undergo fertilization. Look at it very well. See? 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 And see. So this one will have this now. This one will have this. This one have this, and this one have this. So now, this is second figure what? Generation. So you now say that of the first string here now, this one is tall. The second, that's number one. Number two is also tall, because tallness is what? Dominant. Number three is also tall, because tallness is what? Dominant. And number four is now short, because short is double. Do you get that point? So now, if you are now saying, what is the phenotypic ratio? What is shown in them? One person, 1, 2, 3 are tall. So 3. 1 is what? Short. So there are 3 tall people and there is 1 short person. That's the phenotype. What is physically what? Expressed. Therefore, the phenotypic ratio now is 3:1. Phenotypic what? Ratio. That's the ratio of those who are tall to ratio of those itself is 3:1. But if you are not looking at their genotype, the total genes they possess, you now have genotypic ratio. This one is phenotypic ratio. This one is genotypic ratio. So now you see what the difference. This one is what? Tall. Do you understand? But tallness means that this person is homozygous tall. The 2 genes are the same. So tall. This one is tall. But it's heterozygous what? Tall. Because the 2 genes are not the same. I don't know whether you are getting what I'm saying. Then this one is another tall, but heterozygous what? Tall. Then this one is what? Homozygous what? Short. This one is homozygous short. Homozygous short. This. So if you now want to go to genotypic ratio to say exactly, you are now saying exactly what type of gene they possess. This one is homozygous tall. These 2 are heterozygous tall, and this one is homozygous short. So the genotypic ratio 1:2:1. You are now saying the exact type of genes they possess. Beautiful. Then that brings us to sex-linked traits. Sex-linked traits. These are traits associated with one sex more than they are associated with the other sex, because the genes for them happen to lie in one of the sex chromosomes. Example of such sex-linked: color blindness, hemophilia, baldness, sickle cell anemia. Now, we will round up this lecture in the next part. Then we'll now look at the details of sex-linked traits, blood groups, blood transfusion, and the rest application of genetics in everyday life. If you are thrilled by this lecture, join us in the concluding part of the lecture by clicking the red subscribe button. Thank you and God bless you.